In the first trial of its kind, the Cedars-Sinai team showed that delivery of this combined treatment is safe in humans. The findings were reported today in the peer-reviewed journal Nature Medicine. Using stem cells is a powerful way to deliver important proteins to the brain or spinal cord that can't otherwise get through the blood-brain barrier, said senior and corresponding author Clive Svendsen, Ph.D., Professor of Biomedical Sciences and Medicine and Executive Director of the Cedars-Sinai. We were able to show that the engineered stem cell product can be safely transplanted in the human spinal cord. And after a one-time treatment, these cells can survive and produce an important protein for over 3 years that is known to protect motor neurons that die in ALS. Aimed at preserving leg function in patients with ALS, the engineered cells are potentially a powerful therapeutic option for this disease. None of the 18 patients treated with the therapy, developed by Cedars-Sinai scientists, had serious side effects after the transplantation, according to the data. The study used stem cells originally designed in Svendsen's laboratory to produce a protein called glial cell line-derived neurotrophic factor, GDNF. This protein can promote the survival of motor neurons, which are the cells that pass signals from the brain or spinal cord to a muscle to enable movement. In patients with ALS, diseased glial cells can become less supportive of motor neurons, and these motor neurons progressively degenerate, causing paralysis. By transplanting the engineered protein-producing stem cells in the central nervous system, where the compromised motor neurons are located, these stem cells can turn into new supportive glial cells and release the protective protein GDNF, which together helps the motor neurons stay alive. Because they are engineered to release GDNF, we get a double whammy approach where both the new cells and the protein could help dying motor neurons survive better in this disease. Along with Avalos, Robert Balow, MD, PhD, previously a professor of neurology at Cedars-Sinai and now global head. The safety trial. The primary goal of the trial was to ensure that delivering the cells releasing GDNF to the spinal cord did not have any safety issues or negative effects on leg function. Because patients with ALS usually lose strength in both legs at a similar rate, investigators transplanted the stem cell gene product into only one side of the spinal cord so that the therapeutic effect on the treated leg could be directly compared to the untreated leg. The team developed a novel injection device to safely deliver the stem cell gene product, called CNS10-NPC-GDNF, to the spinal cord of patients. After the transplantation, patients were followed for a year so the team could measure the strength in the treated and untreated legs. The goal of the trial was to test for safety, which was confirmed, as there was no negative effect of the cell transplant on muscle strength in the treated leg compared to the untreated leg. We're excited that we proved safety of this approach, but we need more patients to really evaluate efficacy, which is part of the next phase of the study, said Johnson, who is also the vice chair of neurosurgery at Cedars-Sinai. Proving that we have cells that can survive a long time and are safe in the patient is a key part in moving forward with this experimental treatment. While there were no serious side effects, the team found that in some patients the cells went too high in the spinal cord, ending up in sensory R. We are very grateful to all the participants in the study, said Svendsen. ALS is a very tough disease to treat, and this research gives us hope that we are getting closer to finding ways to slow down this disease. The Cedars-Sinai team is also using the GDNF-secreting stem cells in another clinical trial for ALS, transplanting the cells into a specific brain region, ca. They have recently treated the first of 16 patients in the new study, primarily aimed at demonstrating safety but also assessing if there are any effects on hand use over time. Make a difference. Sponsored opportunity StorySource. Materials provided by Cedars-Sinai Medical Center. Note: content may be edited for style and length. Transplantation of human neural progenitor cells secreting GDNF into the spinal cord of patients with ALS. A Phase 1/2 trial. Nature Medicine, 2022. doi. 10. 1038 per second 41591, 022, 01956, 3. Cite this page, Cedars-Sinai Medical Center. 2022, September 5. Stem cell gene therapy shows promise in ALS safety trial. Science Daily. Retrieved September 5, 2022 from ift.tt/7 Wixaka Cedars-Sinai Medical Center. Thanks for watching. Please subscribe my channel for more videos.